All right, this is part 2 of cellular organelles, or lysosomes, peroxisomes, and vacuoles. Oh my! Lysosomes, peroxisomes, and vacuoles. Oh my! Well, trying to make it interesting, but may not be. All right, so let's look at this. First of all, we've covered several of these organelles already. Here's the nucleus over here. Oops, let me change that. Here's the nucleus over here. You can see the nuclear envelope with the double membranes. You can see the little nuclear pores where the RNA comes out. You can see the rough endoplasmic reticulum here, the smooth endoplasmic reticulum, the Golgi apparatus. See a few vesicles here. Now we're going to talk about another organelle called the lysosome, which is actually a pretty interesting guy. Not very much to look at, but really kind of an interesting organelle here. The lysosome is simply just a sac that's full of hydrolytic enzymes, and these enzymes can break down basically anything. Some cells can use them to digest food. All of our cells use them to break down worn out organelles and recycle the parts. What's interesting about these lysosomes is that since they're full of such nasty stuff, the cell needs to be able to control when the lysosomes are actually are active or not. So let me show you how that actually happens. The lysosome has a double layer phospholipid protein studded membrane like that. But then the lysosome also has in it, some proteins, so little protein studs, which we'll make little red studs like this. Those little guys right there are responsible for grabbing up hydrogen ions, which will be out here, H+ ions, and actually bringing them in to the lysosome. In other words, what they can do is they can lower the pH that would actually be inside the lysosome. The reason that's important, two things. First of all, because the enzymes that are actually inside the lysosome are activated by low pH. In other words, their optimal pH is more acidic. That means that when the lysosome's working, it's going to want to bring hydrogen ions in. The second thing that's kind of interesting about that is the fact that how those hydrogen ions get in, and that is that this membranous protein here is actually a transport protein. So these little transport proteins are going to take these hydrogens from outside here, where they're in low concentration, and move them inside so they're in higher concentration. And that's going to require some energy. And of course, the energy we're going to see is going to be ATP. And what will happen is that ATP will be grabbed up by that transport protein, and the terminal phosphate will be broken off. So now we'll get ADP plus P sub I. So this is the first place that we've actually seen the use of ATP to do something. And essentially what we're doing is we're using that energy, that potential energy that was in ATP, to move those protons inside and increase the acidity inside the lysosome. All right, so here's how these guys get made. They're just proteins, so they're made the same way. There's a messenger RNA molecule, which is a transcript of the DNA in the nucleus. It comes out, it's picked up by one of these ribosomes, and then the ribosome makes a protein into the lumen of the ER, which then pinches off into a transport vesicle, which then goes to the Golgi apparatus. But this time, the Golgi apparatus tags this thing not for secretion or to leave the cell, but to actually make a lysosome. So these little guys that pinch off the Golgi apparatus here are now lysosomes. And you can see the function here, and that is that the lysosome can merge with a broken down organelle. This is a broken down mitochondrion, and it can actually break that down and recycle the parts. Or, in this particular case, you can see the cell doing another transport trick here, which is called phagocytosis, an unfortunate pronunciation, but still phagocytosis, where it invaginates or engulfs a food particle there, putting it in a food vesicle, which then is merged with the lysosome so it can be digested. And what this means is that the cytoplasm of the cell actually isn't subjected to these hydrolytic enzymes, that it can actually be sequestered from the rest of the cell. And what they don't show here is that when this is all done, the lysosome will actually break back out of this, so you get your lysosome back, and then now you have a waste vesicle which can come down here and merge with the membrane, and it can excrete all that stuff out. And by the way, the difference between excrete and secrete: excrete means to get rid of waste products, secrete means to give off a useful product. All right, a couple other things, and we're really not going to cover much of these. One you've heard of is glyoxysome. This was earlier in the year. This is what seeds use to break the fats down into carbs so the seeds can germinate. But there's another organelle called a peroxisome. And this guy contains catalase, which is the enzyme that you saw when we worked with yeast. I want to show you a picture of the peroxisome. It's extremely bizarre. Inside the peroxisome here, you see this crystalline lattice of molecules. And I just think it's really weird. By the way, the green color is totally artificial. All right, so now we're going to go and look at some things that plants have that animal cells don't have. And they have large vesicles, large membrane-bound sacs They often have a central vacuole, and here's your typical plant cell. You can see this large central vacuole here. The membrane of that's called a tonoplast. And then we're going to see some protists, which are going to have a vacuole which pumps water out of the cell. We'll talk more about that later. And by the way, just so you see, a vesicle is a small membranous sac, and a vacuole is a large membranous sac here. Okay, so here's again a highly colorized version of a plant cell. You can see the membrane with the cell wall, the nucleus here. These guys are chloroplasts, and you've got a really, really large central vacuole with the tonoplast. Once again, plant cells can in general be much larger than animal cells because the central vacuole doesn't require a lot of metabolic activity. So by having this large central vacuole, the membrane only has a surface this much of the cytoplasm. The central vacuole is used for lots of things. It's used to store things like sugars. It can be used to store waste. It can be used to store colored pigments, and this gives plant parts their different colors here. The next organelles are quite bizarre in the fact that they really are kind of like little cells that live inside of other cells. And we're just going to look at their structure in general now. We'll talk a lot more about these guys later on. We talk about respiration, photosynthesis. The first one is the mitochondrion. The plural of that, by the way, will be mitochondria. And this is the site of cellular respiration, aerobic respiration. This is where the organelle is going to take the glucose plus oxygen, and it's going to break it down to release that energy. And of course, that energy is used to make ATP. It's a really weird organelle in the fact that it has an outer membrane and then it has an inner membrane. And what you'll notice is that this inner membrane has these tight folds called crista, cristae for plural. And these cristae obviously increase surface area. And there's all kinds of proteins in this membrane. So that folding of the membrane increases surface area and increases the number of proteins here. There is an intermembranal space, and then there's the inside of this inner membrane is called the matrix. Now, what this really looks like, electron micrograph. Again, the color is just because the guy that did this thought red was really pretty. You can see the folds here of the cristae. You can see the double membrane and so forth. Another organelle which is similar in a way and actually does kind of the opposite thing is the chloroplast. You've already seen some chloroplast in Elodea here. This is the organelle that carries out photosynthesis, CO2 plus water in the presence of light, and this pigment chlorophyll makes glucose and gives off oxygen. Now this guy actually has three membranes. So let's take a look at a chloroplast here. So what it's got is it's got this outer membrane, and then it's got an inner membrane here, and then it has a third membrane which is highly folded back and forth. The little inner membranes here are called the thylakoids, and that's we'll talk more about those later. Again, there's a stroma, which is the inside of the chloroplast, but outside of the thylakoids, inner membrane, outer membrane. And there's some notes on this. Again, this is an electron micrograph, colored green, of course, and you can actually see the structure here on this. If you'll notice that the chloroplast and the mitochondria have their own DNA and their own ribosomes, and they are self-replicating. So again, they are very much like little cells inside of a cell, and we'll talk more about that right now. Both of these mitochondria and chloroplast are thought to be the result of a process called endosymbiosis. And in other words, at one point they were cells that were living independently and got engulfed or phagocytized by larger cells, and then actually weren't digested for some reason, but it turned out to benefit both the organelle and the cell itself. This is one of the types of interspecific or interactions between species called symbiosis. Symbiosis occurs in three forms. Everybody's pretty much familiar with parasitism. That's where the host organism is harmed and the parasite is benefits. There's also mutualism, and that's where both organisms living together benefit. I'm reading a book currently about some leafcutter ants, and the leafcutter ants cut leaves and feed it to a fungus, and then they eat the fungus, so the fungus is benefited because it gets fed, and the ants are benefited because they get fed too. And there's commensalism, and this is where one species benefit, but the other is neither harmed nor benefited. This is perhaps the clownfish and the sea anemone. The clownfish gets protection, but nobody's quite sure what the anemone gets out of it. So endosymbiosis is the idea that certain prokaryotic cells actually got engulfed and became organelles. And as time passed, they became what are known as obligate symbionts, and that is that they now cannot live apart from each other. The evidence for this are the fact that both mitochondria and bacteria are surrounded by double membranes, which the second membrane would be the one where they get engulfed. They both have their own DNA, and ribosomes are self-replicating. In other words, they don't depend upon the DNA of the nucleus to actually replicate. They divide just like bacteria, and the DNA is both the DNA in both is circular, which we haven't talked about this yet, but in prokaryotes, bacteria, we see that DNA is in a circular form, not linear. Okay, that's going to do it for now. We'll continue this later.